Thank you so much for the organizers of this meeting and you all for coming. So we are excited about the possibility of malaria challenge because malaria is a big global health problem, and for DREAM this will be the first infectious disease challenge. And even though malaria has been studied for many years, one of the challenges in the disease is in terms of developing drugs that can be robust to emergence of resistance. So one interesting thing is that the very first drug for an infectious disease was actually an antimalarial, quinine. That was a very successful drug for 400 years. Surprisingly, in the 1940s, a new drug, chloroquine, was discovered, and chloroquine was only successful for about 10 years before resistance began to emerge. And then from then on, every other antimalarial drug that has been introduced, we have seen resistance in less than a decade. So why is this happening? If anything, shouldn't we have better medical discoveries today than we did 400 years ago? Why was quinine successful for 400 years? And why is it that every other drug we discover for malaria today is active for less than a decade? So artemisinin is another antimalarial drug that is viewed today as a very important drug in the eradication of the disease. It's used as the first-line drug against the disease, and actually a Nobel Prize was awarded for the discovery, or should I say, for the characterization of artemisinin, because artemisinin was used for centuries by the Chinese as a herbal medicine. But the problem we are beginning to see, even with artemisinin, is that resistance is beginning to emerge, especially in Southeast Asia. There's another complication about the emergence of artemisinin resistance, because it's not a typical form of resistance like the resistance we've seen against chloroquine and other drugs, in the sense that the current phenotype of artemisinin resistance that is seen in the field is a phenotype that is associated with delayed clearance of the parasite from the patient bloodstream. So if a patient continuously takes the drug can still work, but you have high cases of treatment failure where it takes a very long time for the parasites to get cleared. And there is increasing fear that the spread and further development of this resistance may curtail the great advancements that have been made towards eradication of malaria. So there is a huge effort to look for new compounds against malaria. So artemisinin resistance in the field has been found to be associated with a mutation in a gene called K13, or the Kelch protein. And there's still a lot that we don't understand about this resistance. So for instance, we know that even some of the parasites that have the K13 mutation are not resistant to the drug. And you also find cases where, like, wild-type parasites that have no mutation show some resistance to the drug. So in other words, there's still a lot we need to learn about the resistance of artemisinin. And we have to remember also that it's an evolving resistance. So we don't know what the endpoint is going to be. So there's a window of opportunity to do something about artemisinin resistance before it is too late. So this is why we think that a Dream of Malaria challenge will be important for the community. And again, it might be a way for exploring new models for research on infectious diseases. And also, I think it would be an opportunity to attract talent and new ideas into how we model infectious diseases, because we've seen a lot of progress in how you model other diseases like cancer, but maybe not so much in the infectious diseases. So this might be an opportunity to do that also. So in 2010, I spent some time at the lab of Gustavo at IBM as an intern. And one of the fantastic experiences I had there was to see Gustavo organize the DREAM challenges. And this is how we came up with this idea that what if we could apply the same kind of idea to the area of malaria? So when I went back to campus where I was a student at the University of Notre Dame, we wrote a grant that was eventually funded by the NIH. And one of the aims of the grant was to generate data that would be used for a DREAM challenge for understanding malaria drug resistance. So the other unusual thing we did in trying to think about how to make this challenge come true was also to try and engage a wider community. So I'm from Kenya originally, and I strongly believe that the future of modeling many diseases should occur on a global scale, and importantly should involve also those who are affected by the diseases in question. So because of this ideal, we decided to organize a hackathon in South Africa, in which we worked in collaboration with a consortium called the H3 Africa Bioinformatics Network. And basically the idea of the hackathon was that we would bring together several young scientists across Africa, bring them in one room. They stayed in one hotel. They were sitting together in the same room for a period of a week. And the IBM Research Africa Lab offered their facilities there. And basically within the week, the young scientists were able to analyze some of the data, learn about how to do the data analysis, work alongside some of the IBM researchers. And also we abstracted the problem in a way that many of the young scientists who had no experience in biology, they could also make a contribution in this. And we also published a paper summarizing the idea of using this kind of setup as a means of training young scientists. So in terms of the DREAM challenge, which we hope to be the next set of challenges sometime early next year. So part one of this challenge is going to be to predict the IC50 of a set of parasites given gene expression data from those parasites. So for the training set, we have a small set of 20 parasites from Southeast Asia. And for each of these parasites, we will have gene expression data obtained at 2 time points, both in the presence and in the absence of the drug. And Sage Davis from the University of Notre Dame has collected most of this data. He'll present a poster also today, so you are welcome to go to the poster to learn more about the challenge. In the second part of the challenge, the goal will be to predict the clearance rate of artemisinin given the gene expression data. So the clearance rate is a phenotype that is measured from the patient. So the level of parasites in a patient who has taken artemisinin is measured over time until when no parasites can be detected in the blood, and that gives the clearance rate. So in the second part of the challenge, the goal will be to predict the clearance rate of the parasites given the gene expression data. So Sage Davis will present a poster that will give more details. And again, we plan that this challenge will be early next year, depending on the schedules of the DREAM challenges. And the work is done in collaboration with several people. So the samples are coming from Southeast Asia, and a lot of the lab work is done at University of Notre Dame. So thank you, and maybe I'll take some questions.
Questions? Can you get sort of a graph?
Thank you. Very exciting challenge. I might actually participate. Is there any reason why you have fewer samples in the training set, more in the test set? Because that would, that would probably hamper developing more robust and accurate models.
So you mean for the IC50?
Yeah. I mean, usually it's the other way, right? I mean, it doesn't have to be, but I'm just wondering if there is a reason for setting it up this way.
Yeah, so there's, I would say there's no particular reason, unless Sage, do you want to comment? Some of it might have to do with the nature of the data. Sage, do you want to comment, maybe?
No, there's no reason why we will have to have less training.
I thought it was a typo. It was 35 training and 20 tests.
Okay, that's technical. Yeah.
Can you put the other slide? Can you put the... Right, so it has to do actually with the fact that, so there's a difference between clearance rate and IC50, and this is one of the important questions, is why the IC50 in vitro doesn't correlate with the clearance rate. And so for the clearance rate samples, we have 35 isolates, and we have a large training set because that's a published paper in Science. And so we just wanted to have a larger test set to have a better assessment of the models, given that for the other question we have a lot of data. So it is trying to equilibrate both challenges. But yeah, it's a good observation. But part of the idea is that 20 was enough to have some predictability. So we prefer to equilibrate both sub challenges.
Yeah, because one thing I didn't mention that, so some of the parasites have both IC50 and the clearance rate, but some of the parasites don't have clearance rate, because the problem is that the clearance rate has to be measured in the patient. So if we have a parasite in the lab that the patient was not characterized in time, then we cannot do anything more about the clearance rate.
Okay, one more quick question.
A question, is this going to be a Docker-based challenge, or you're going to provide the predictor for the test set directly?
Well, so we are not planning to use Docker at this time, but something to think about in terms of also enhancing reproducibility, and so definitely.
Okay, let's thank Geoffrey again.